Hello, E3 Scholars. Welcome back to Sharp Shorts. Now, we've looked at some at tutorials that talked about the cell cycle, and we specifically looked at mitosis to keep the integrity of the DNA there. On this film, we're going to look at what is the triggers, what is the pacing, what are the mechanisms, how does the cell cycle know when to do what it does. There's one theory, very simplistically, just says you have G1, you have S, you have G2, and they just follow sequentially in order, and one follows the other, and that's not it's a plausible theory. The problem with it is that it doesn't lead to, if it's just a straight metabolic pathway, it doesn't lead to any triggers from the environment, internal or external. And that's a difficult thing to correlate with multicellular organisms and plants and stuff that have different cells on different cycles, and what controls them, what triggers them. So really, as you can see by the experiment listed here and in your book, it must be some determinants, some chemical, it has to be chemical determinants in the cytoplasm of some sort, because as you can see here, if you had a cell in the S phase fused with a cell in the G1 immediately went into the S phase. So there was something in that S phase cytoplasmically that triggered it going right into S phase. And correspondingly in experiment 2, if you put a G1 cell, fused it with something in mitosis, that also then immediately began mitosis, even though the chromosomes hadn't been duplicated. And so there was something, and that is of the intense interest, that would be studied and looked for to see what is the controlling mechanism. Okay, so the events are kind of what they like to equate it to, and I'll get to the picture here, is like your washing machine doll. There's distinct events, like the wash and the rinse cycle and the spin cycle and everything else. And these distinct events, if you kind of wanted to believe it was like a cycle, they go, but they have checkpoints. Before I do this, do I have enough water in the bin? Do I have this and that? So what you can see here are the phases of the cell cycle, and you can see where key checkpoints are. And I'll tell you right now, G1 is kind of the key checkpoint. And this is, as we talked about before, right after mitosis, you have that growth phase. And when you get to that checkpoint, it's am I going ahead or am I staying here? And oftentimes, like neural cells, when I get here, I'm shunted off into a G0 phase, it's called, which, and a lot of cells are. A lot of cells are in the G0, G1 checkpoint because they just don't continue rolling through mitosis at a rapid rate. That doesn't mean they're dormant, as we said before. It just means they're going about doing normal cell stuff. The key to this is once it does pass the G1 checkpoint, it is going to divide at some point, because it's going to go through copying its DNA. It's going to go through the G2 phase. Now there's a checkpoint here at G2 to make sure it's ready to go, and we'll look at that pacing and the things that control that. And then again, there's a mitosis checkpoint, which is that metaphase checkpoint. Is everything lined up on the metaphase plate, and are the spindles attached to the kinetochores? What is that pacing? And then again, it's the go-ahead, non-dividing state, the G0 phase. What is that pacing? You can see here again, am I going to go or am I going to be shunted into G0? Well, the pacing of it seems to be dependent on what are called cyclins and cyclin-dependent kinases. Okay, and that activity that fluctuates, and you'll see a thing in a minute. The kinases are generally there at a constant level throughout the cell, but they need to be, but they're inactive. Okay, so the CDKs are essentially at a constant level, but it's the fluctuation of the cyclins. And it's their level and their production, and the CDKs need to be bound to the cyclins to be active. Okay, once they are active, and one of the first ones discovered was called maturation promoting factor, some call it the M phase promoting factor, is the one that triggers the cell past G2 into the M phase. And here's a good illustration of it. And you can see what happens here in the M phase. You can see the cyclin concentration in the MPF are high. Now what I'll tell you, what's shown here is the CDK is essentially constant through, as you can see on the circular diagram here. But it depends on the cyclin building up through the phases to trigger it, and then when enough of that binds up, then you increase the maturation promoting factor, and when you get enough of that, then you go past. So you can see the fluctuation in the cell cycle depends on the cyclin concentration, and that is a pacing guide, a pacing control molecular mechanism that helps go through. The nice thing about this kinase and this MPF function is it not only triggers once it builds up, and once you build up enough and it triggers past the G2. This is also, there's other kinases and stuff, triggers other kinases to dissolve the nuclear membrane, etc. It also triggers its own destruction at anaphase, in that it starts degrading the cyclin so that level goes down, essentially inactivating the CDK, so that when it hits G1, it's done, and it's at that low level, and then you're on the pacing time again. Okay, and that's not the only one. They've discovered a number. This tells you G2. There's at least 3 that control past the G1 part. Now, what are external and internal signals that can, that also control and trigger? Because again, as a multicellular organism, you have different cells going in different times. You have repair mechanisms that need to occur if cells are damaged. Okay. We talked about the M phase one. The kinetochore is not attached to the spindle microtubule, send a molecular signal that will delay anaphase. Other external signals are growth factors, and those can be a few different things. Density dependence on the cell. Cells grow until they essentially touch each other and are a single layer thick, and then that will stop. Or other triggers. Platelet-derived growth factor, as illustrated here. These cells are not going to go without what's called platelet-derived growth factor, which you can see here by the experiment and read about in your book. There's no growth, even though they're in the same nutrient media. These fibroblasts that are there to differentiate during repair and stuff, this is all part of the blood infection and response. If there is wound repair and damage here, these fibroblasts will not differentiate and start the repair process of the cells without the platelet-derived growth factor. So that's an external control that when the repair signals goes out, that would be something that would then bind and cause triggering and maturation. Okay. Here's another one, making new red blood cells. Erythropoietin is a hormone that is kicked out when you need more red blood cells, and they would bind externally and trigger. Growth factor erythropoietin made by the kidney travels in the blood to bone marrow. Okay, and when it binds, then it will trigger that we need more red blood cells, and of course it's a hormone factor, so there are things that would trigger that also in the hormonal system. But anyhow, the whole idea is there are internal and external factors and molecular control mechanisms for the pacing that guide this. Now, we talked about density dependence. There's also for animals anchorage dependence. We're one layer thick, and we need to be stuck to basement submembrane to go. So how does cancer occur? Well, there's numerous ways of cancer to occur, mutation and everything else, but what are some of those signals that we can look at that says they're not there? And density dependence and anchorage dependence are things that cancer cells don't rely upon. You can see here one of those external signals. Cells will grow until they're a layer thick, they're touching each other. That side touching tells them to stop. That's an external control to control the checkpoints of the cell cycle. You scrape away some of the stuff like here in the micrograph or here, and it will trigger again here growth until that fills in. cancer cells not only don't follow that density dependence, but they also are not necessarily, as you can see, they're piled up here, but they also don't rely on anchorage dependence where they would be bound to a tissue right below. And that's just a couple. Really, the idea is, for some reason or another, cancer cells don't need the same growth factors, don't need those triggers to grow and divide. They may make their own once they mutate and become cancer cells. They may go ahead and go through without the growth factor there. They may have abnormal cell cycle control systems, and some, again, this is part of the difficulty in finding a cure, quote unquote, for cancer, is what are those triggers and stuff that they're doing. Right now, one of the key researches is not looking so much at the cell cycles, but the abnormal proteins that are there, the things like that, and attacking those proteins, which then seem to get a more normal response back to it. Anyhow, normal cells that become cancerous go through by transformation. Cancer cells are not eliminated by the immune system, form tumors, and that's just unchecked mitosis, basically. If that abnormal cells in that lump of cells, that non-density dependent, stays in the site, is still kind of anchored, then that's a benign tumor. It's when it loses that anchorage dependence and runs through the lymph system, then into the blood system, that they're called malignant, and they can, as they start moving into the surrounding tissue, and then they can metastasize and go through those delivery systems in the bodies where they may form additional tumors. We have this example here. You can see that you're getting a tumor here from a single cancer cell, but it starts invading not just the glandular tissue, but the epithelial tissue, connective tissue, and everything around there, and then losing its density dependence, and whatever triggers it was that it's blowing right through the cell cycle controls. And then when it loses its anchorage dependence, it will go through the lymph and blood system, and it can metastasize in other parts of the body. Anyhow, so that gives you an idea of how important those cell cycle controls are, and kind of the breadth of what can happen with cancer. Very difficult with all the different cells and the different types of things that goes on to understand, but at its basic, they are missing some of those cell cycle controls and pacing guides and stuff where they have thwarted them in some fashion. Anyhow, that gives you an idea of the controls. Scholar up, Eastview.